Hello, welcome you all. Today's session is about laboratory facilities. I am Sanjota Ji. I am going to explain about laboratory facilities required for the organization of plant tissue culture. In previous session, we have discussed about history and development of plant tissue culture and plant biotechnology and what are the tissue culture techniques used for in vitro cultivation methods. The laboratory setup for tissue culture depends on the nature of research and the availability of funds. Even though simple experiments are performed in this laboratory, but synthesis of secondary metabolites and genetic manipulation, those also can be carried out in tissue culture laboratory. Let us discuss what are the basic facilities for organization of a tissue culture laboratory. First and foremost thing is washing and storage facilities. Later, cleaning of glasswares. Sometimes we use plastic labwares. Those also should be cleaned. Later, media preparation room. Media preparation room is very important, and what all equipments are equipped in media preparation room, that we will discuss. Next facility which is required by the organization of plant tissue culture lab is transfer area. Next one is culture room, data collection area and specialized facilities, and transplantation area. Let me illustrate one by one. The main requirement for washing and storage is an area sufficient for the accommodation of large sinks and provision for the running water and draining boards. Distilled and double distilled water operators must be installed in this area. Drying ovens must be kept. Washing machines should be there. Detergent pots, dryers, cleaning brushes and dust-proof cupboards must be there. A tissue culture facility requires continuous running cold and hot water facility, and drainage of disposal water is must. Disposal of any waste is governed by local municipal codes and international norms to minimize health risks and environmental hazards. There must be cleanliness is maintained in laboratory setup. The second facility which is obligatory for lab setup is cleaning of glassware. A good quality glasswares are recommended for this purpose. For example, borosilicate glasswares. Why? Because we use high temperature for sterilization purpose. In hot air oven, 160 degrees centigrade we are using, and in pressure cooker, 120 degrees centigrade is maintained. This much high temperature is recommended in laboratory. So the glasswares which we are using for this purpose must be resistant to more heat. The conventional method of cleaning of glassware is soaking of glasswares in sodium dichromate and sulfuric acid solution for 4 hours. After 4 hours, we have to wash all the glasswares under jet of tap water. Instead of using acid for washing of glasswares, we can also use detergents. If we are using detergents, we must soak glasswares in detergent solution for 16 hours. After 16 hours, we must rinse those wares in tap water and second rinse in distilled water. After distilled water wash, we have to dry those glasswares in an oven at high pressure and temperature. We can use 150 to 200 degrees centigrade for the drying of cleaned glasswares. Plastic wares may also be used in the laboratory, and these may be washed with a mild non-abrasive detergents. After washing with detergent, we must rinse with tap water, later with distilled water. Then those plastic wares and cleaned glasswares can be kept in dust-proof cupboards, and whenever we need, we can use those glasswares for the tissue culture experiments. The third facility, which is mandatory for lab setup, is media preparation room. Here, ample space must be there for keeping chemicals, labwares, and culture vessels. Some of the equipments which are required for media preparation must be there in this room. Provision for placing hard plates, pH meters, and balancers. And micro oven should be there, autoclave must be there, culture vessels, instruments which are needed for the media preparation must be present in the media preparation room, and vacuum sources, refrigerator and freezer for storage of some chemicals which will get spoiled at room temperature. The next facility is transfer area, where all the experiments or inoculation is done in this area. For this purpose, we can use small plastic box or wooden box that must have UV light and fluorescent light inside. This is used only for simplest type of transfer. The plastic box or wooden box must have sliding door, and the door must contain two holes to insert our hands, and we can perform the experiment. For large number of cultures, we must use large rooms with an overhead UV and positive pressure. Large room must be sterilized by using some chemicals, and the large room must have UV light. Ventilation unit must be there, and which should possess high efficiency particulate air filters. Currently, most of the laboratories use laminar airflow. The most important area is culture room. After inoculation, we bring all the culture tubes and bottles to this room, where the growth of the plantlets will take place. Hence, the culture room must be equipped with all the necessary factors. Well-controlled temperature must be there, humidity and illumination and air circulation. Here, when we bring the test tubes or inoculated bottles, then those are kept in the racks. The rack or each shelves must be equipped with fluorescent light, and at one end of the shelf there must be presence of small fan, and that fan is fitted with tube which is having uniform sized holes. When fan is connected to electricity, then the whatever the sterilized air will comes from the fan will enters to the pipe, and each and every plant tissue culture tubes will get the uniform distribution of air. So that facility must be there in the culture room. Temperature which is used in this area is 25 degrees centigrade plus or minus 2 degrees centigrade, and light intensity less than 1 kilolux, and humidity must be 20 to 98%, and specially designed shelves illuminated by a set of fluorescent tubes must be there in this culture room. Next facility is data collection area and specialized facilities. In this area, we have to collect the data of the culture tubes. After inoculation, we must note down whatever the nutrients which we are adding and what temperature we are maintaining, what is the humidity, what is the pH we have maintained. All those information must be noted down, and after transferring the culture tubes into the growth chamber or culture room, we must note down the date of the inoculation. Later, after 1 week, we must see whether any growth takes place in the culture. Then the growth and development of tissues are monitored at regular intervals. For germplasm conservation, must provide lower temperature rooms and cryopreservation units. These are the specialized facilities which must be there in the data collection area, and the growth and development must be taken regularly. This is most important facility which must be there in the laboratory, that is transplantation area. Here the acclimatization of tissues or plantlets will be done. Acclimatization, which I have already explained in the previous session, that is hardening of the plantlets. After plantlet growth, the plantlets which are grown in laboratory condition are very sensitive. They must be adjusted to wild condition. Hence, before transferring those into wild condition, the plants regenerated in vitro are transplanted to soil in pots. Sterilized soil is used first in sterilized pots. Later, potted plants are transferred to greenhouse under controlled light, temperature and humidity condition. After greenhouse effect, then the plantlets are able to adjust to the wild condition. Here are some pictures. You can see this is laminar airflow, and she is working with some of the culture tubes. This is the laboratory or transfer area. This is the plant tissue culture laboratory. Here many racks are there. Each shelf is fitted with fluorescent light, and some shelves like they are specialized, they are fitted with fans. And this is the rack where cultures can be kept. And this is greenhouse effect, greenhouse area where plantlets are undergone acclimatization. These are some of the references which I have used for the preparation of this session. Thank you.